All right. So, thank you, Laura. I'm Yossi, indeed. As was said, I'm here for a few more weeks, 6 actually, after this workshop. So if you're around and you have more questions, feel free to come by. I'm sitting at FIM in Biomedicum 2. And also, despite the request that you guys be quiet, that should not preclude anyone from asking any questions. Feel free to interrupt, raise your hand or shout a question or whatever. I'm happy to respond to any questions that you may have in the middle. Okay, so this is about how our data gets generated. The first part, the part that Laura was mentioning, happens at the Genomics Platform. So basically, we have samples coming in. Usually, they either are or are not. The DNA has or has not been extracted from the cells, and we need to turn that into data. So we have 2 main types of data that we are generating. One is whole genome sequencing. So whole genome sequencing, we basically, as it says, take the entire data and the entire sample, and we extract reads. We read everything that we see there, and we can find variations that are either in exons or in introns or even intergenic. We will find reads over there. The whole genome sequencing has very little bias with regards to different parts of the genome, and so that enables us to find variation everywhere. Of course, people know mostly how to interpret variation when it happens in a genome, in a protein coding region, so over the exon. But interpretation is one thing, and finding variance is another thing. If we can't find variance, we won't learn how to interpret them. So the downside with whole genome is that until recently it was extremely expensive. So the genome is the entire genome is about 100 times larger than the exon and so if you up until that giant increase in sequencing ability that Laura was showing in the plot in the graph just a few minutes ago, until those new machines came in, it was a lot more expensive to do whole genome than to do exome, and so people were doing exome. So what's exome? With exome, we simply extract the reads, the parts of the DNA that overlap with the exome, with the exons in the genome, and we read these. So we get a lot of reads over here, and very few, if any, reads in between in the exonic region and the intergenic region. Okay, so that enables us to find variants here, but we are blind to whatever is happening here. So when we take our DNA, we could take DNA or RNA. If we take RNA, we reverse transcribe it to DNA. We chop it up into fragments because the sequencer can only read fragments of size around between, say, 100 bases and 1,000 bases. So we chop it up into about size 400. That's our kind of target for fragment size. And then various lab protocols add these adapters, they're called, but you should think of them as handles, right? They're made out of DNA, but they fit the sequencer, right? So they fit the sequencer in a way that enables us to sequence them. And so these adapters, we add them at the end of the fragment, and that enables us to put this on the sequencer. It sticks to the sequencer, and then we can read it through. It also has these primers that help us begin the process of sequencing. So that's library preparation. Looks like there's very few steps here, but it's actually quite a complicated issue, and you have to perfect that in order to reach very little bias and all those issues, because of course, because it has to. This is DNA, so things like GC bias and the length make it so that if you don't do it carefully, you only get coverage in parts of the genome and not other parts. Once we have this library, we put it on a piece of glass that is called a flow cell. This piece of glass has these channels etched into it. It's actually it's not on the glass, it's in the glass. So these tunnels go in the glass, and the inside of this flow cell is coated with material, with DNA, with short strands of DNA that exactly stick to those handles that we were talking about a second ago. Okay, so the DNA sticks to this flow cell, and then by process called bridge amplification in the newer machines, other types of amplification in it's always bridge amplification, but there's different types. In the older machines, we get these clusters of coherent strands of DNA. So each of these clusters will have the same piece of DNA replicated many, many times, right? And then by this process called sequencing by synthesis, we read one base at a time. So we add a base to all of them. We photograph what we see. Every base has a different color. So we get a bunch of color coming back, and we can tell from the color which base is there. Then we do a chemical reaction. We get the next base on, and the next base, and the next base. And every time we take a picture, we see all of the images. We see all of the bases for all of the millions or almost billions of molecules that are being sequenced at the same time. And then we build the sequence of bases one cycle at a time. So each time we put on a base, it's called a cycle. And then we flip the strand over and sequence the other side. So if we see the fragment, it's got adapters on both sides. We can sequence one side, and then we sequence from the other side. Okay, so we get two ends of the same fragment. All right. So once it's sequenced, we emit this data that includes the sequence name. So the name tells us some information about where this read was found on the flow cell, and what the name of the flow cell is, and the name of the machine, and so it's physical attributions of the read, and then the actual sequence, and then something that's very important, and we will talk a lot more about later, which is the associated quality score. So every base has a quality score that says how accurate, how confident the machine is in the quality of that, in the value of that base. Right? So if it says it's an A, well, how sure is it that it's an A? Is it like 50-50, or is it, more than that, right? So so here is an example. You see the name of the read, you see the bases, and then you see qualities. So these qualities are coded. This is ASCII. So the ASCII code, the number that this corresponds to, translates to a Fred-scaled Q-score. Fred-scaled Q-score is basically a log scale. So it's minus 10 times the log base 10 of the error. Okay, so what does that mean? It means that Q10, if this equals to 10, it means that epsilon here is 0.1. So 0.1, this will be minus 1, and then times 10 is minus 10, and then minus is 10. Right? So 0.1, so the probability of an error is 0.1, 10%. Then you get Q10. So Q10 is not so great, right? 10% of an error, that's quite high error. If I say something is, an A and I have 10% error, well, how much can you trust that, right? I'm going to give you 20 bases. If all of them are Q10, then 2 of them are wrong. That's not really good, right? So Q10 is not that great. Q20 is starting to be reasonable, right? So Q20, what does that mean? It means 1% error, right? So if this is 20, minus 20, minus 2, so E is 0.01, right? Epsilon is 0.01. That means log 10 by 2. Yes? All right. And then Q30 is 1 in 1,000, right? So here's a kind of a plot to exemplify what that means. So as we go from 0 to 60, the error, the accuracy shoots up from 0 to 100%, and the error rate goes down from 1 to near 0. And as you can see, once you're above 20, then you can kind of say, well, this is pretty good quality. Right? So this is also how we think of it Q20 is our threshold for many things regarding quality. One important point, and we will talk about that again later, is that these qualities are what the machine thinks the error is. The machine is not always right, and we will fix it as one of our steps. All right. So, what do we need to do when we get the data? We get these raw reads. We need to map them to the reference. The reads don't come with any information beyond, as I said, their name, the bases, and the quality. So we don't know where they came from the reference, and we don't know, maybe some of the steps in the library prep creates duplicates. We don't know whether some of the reads are duplicates, and in that case, we will want to ignore the duplicate reads and only use one representative of the duplicate set. And then, as I said, the quantities have to be recalibrated because the machine is not always right about the quality of the machine of the reads. And then we have reads that are ready to go. Okay, so that's the pre-processing. All right. For mapping to the reference, we use VWA MEM. It's an aligner that we think is currently the best for our purposes. Of course, for RNA, you might want to use something else that takes into account the gaps that could happen in RNA reads. So we use STAR for that. And once we map, that means that every read has a place in the reference, and we can draw it like this rather than drawing it like just a big jumble of reads. Right? So the reads kind of they are coherent with each other over every piece of the reference. Here is what a BAM looks like when it comes out of the mapper, the aligner. I'm not showing a header that contains all kinds of meta information about the file, but each record looks like this. If you look at a BAM file, you will see this. You'll see the read name, the same read name that was emitted originally. You see a bunch of flags, which each binary piece of this number tells you something about that read, and you could figure out what that means in a website called Explain SAM Flags, which should be mentioned here, but it isn't. This is the position where it's aligned to, the start position. So it's chromosome 1, position, what is that, 10,052,169. This is the mapping quality. So the mapper also says how confident it is that this is the place that the read came from. Mapping quality 60 is the highest quality that BWA MEM gives. And it could be mapping quality 0, which means that it actually has no idea where it should go. It could either go here or somewhere entirely different. Cigar string we'll talk about more later, but it basically tells us how the read is actually aligned to the reference, right? Because the read might not be exactly correct in place. There might be some insertions or deletions or parts that the aligner says, actually, this doesn't fit at all. So all that information goes into the cigar string. Mate information, remember the reads came in two sides, read 1 and read 2. So the mate information tells us where the other mate is aligning to. So we have the equal sign means it's the same chromosome, that's good. It's not the same chromosome, it means that something is strange is happening, right? If read 1 is on chromosome 1 and read 2 is on chromosome 15, they came from the same molecule, and so either there's this strange fusion going on between chromosome 1 and 15, which is possible. In cancer, everything is possible. Or it's a part of the genome that doesn't map very well. That's also possible. Or your sequencing quality is very bad. Or your data prep is very bad. There's a lot of things that could go wrong. If that's happening a lot, that's usually an indication of something strange. So same chromosome. And then the position. I find it hard to believe it's 14, but anyway, position 14 and mapping quality. Okay. I think these were shortened. I think these were manually edited to make it fit in the page. Read sequence and quality scores, again, as we talked about before. And then there's a whole slew of metadata that tells us information about the read, and it's non-structured. So this is basically a dictionary of tags and values, and there's a documentation about what the different tags mean, but it's changing all the time. And so this is structured here, and then this part here is less structured. It's still, there's a specification, but it's not as structured. All right. So two more things that need to be explained. One is the position, and the other is the cigar. So it's the position. Here I said this is the position of the alignment. This is the starting position of the alignment, and there's something slightly confusing because, let's talk about the cigar first. So the cigar says, so there's a bunch of different operators and numbers here. So you see there's a 1S, 3M, 1D, 2M, 1I, 1M. So what does that mean? S means soft clip. It means the aligner actually thinks that this base is not part of an alignment. It's not a deletion. It's not an insertion. It's just like wrong, completely not part of the read. Maybe the aligner thinks that the insert was so short that the sequencer was reading beyond the original piece of DNA and was reading into the adapter, right? So then it's not an insertion. It's not a deletion. It's just not, right? So that's what we have the S for, the soft clip. 3M means 3 match, so 3 bases that align to the reference correctly. 1D, 1 deletion. 2 more, so 2 bases were, 1 base was deleted, then 2 more bases match. 1 base was inserted, and then another base match. Of course, the numbers here were made smaller because we need to fit into this. Usually the numbers of matches are going to be larger, but just for illustration purposes, we made the numbers smaller. Yes?
You made the information, the second number is not the button of the template length.
Thank you, that's right. That makes more sense. That's a good point. So it also doesn't, maybe I should go back to the, right. So here we're saying template length of 10, which basically means that the distance between the distance between the position of the first read, the position of read 1, the position of read 2 is 10. So here, let's see if we can understand what this means. So this is a cartoon, so we pretend that the reference starts here, and this is aligning at position 2. Notice that the first base is at position 1, but this is position 2. Okay. So the reason is that the first base is actually clipped. And this is somewhat confusing. This is the alignment start. The first read is not, the first base is not aligned. It's clipped. Because it's not aligned, the alignment actually starts on the second base. The second base is at position 2. The second base is the beginning of the alignment. Okay, so this is a slightly confusing thing, but if you don't see that in the beginning, then it's very difficult to understand how things are, why marked duplicates have such a hard time. So one of the reasons that one of the tools will have a hard time is because we're sorting by the alignment start, not by where the read actually starts. So the read starts at base 1 here. The alignment start is at position 2 because we have 1 base that's clipped. All right, so 1 base is clipped. We start at position 2. Then we have 3 matches. And then we have a deletion. That's what a deletion looks. There's just no base here. Right? And then we have another 2 matches. And then we have an insertion. So we put a gap in the reference to illustrate that. Another match. Another confusing thing that should be mentioned, even though it's not illustrated here, is that a match does not mean that the bases agree. It just means that the alignment agrees. So we could have a C here instead of an A. That would still be an alignment match. There is a different operator in the cigar string to illustrate that, but BWA MEM does not emit those operators, and we don't actually use it. So we don't need to talk about it. Okay. At the Broad, we do not use fast queues. The reason is that fast queues do not have all that metadata that I talked about in the header, and they don't have the ability to put that dictionary with extra tags at the end of the read. So we do not use we don't use fast queues. We go from the sequencing data from the machine, we go directly to an unmapped BAM. In the unmapped BAM, we can put all the metadata that we want. So we can write what the sample name is, and we can put information about where the like what the machine was, and all this extra information that we want to keep in the file. We don't want to store that in a database outside of the file where it might get confused and we might mislabel things. So we keep it in the file, all that metadata in an unmapped BAM. Then, in order to align it, we have to turn it into a FASTQ because BWA takes in a FASTQ. So we turn it into a FASTQ and give it to BWA. We never save that FASTQ. Stream it in. We get a mapped BAM. And then but that mapped BAM, of course, will not have any of the metadata because we went to FASTQ. And so we merge it together with the original file using this program called merge BAM alignment. So now we have a mapped and annotated BAM file. We sort it, we get a sorted file. And now our sorted file has the metadata and has the alignment information. All right, the next step is to mark duplicates. So marking duplicates is a way of making sure that that we don't use dependent reads that came from the same molecule. We don't want to use them twice. Our confidence numbers are heavily dependent on the fact that all the different reads are independent. So we need the reads to be independent. If we have reads that are dependent, if we have reads that are duplicates of each other, we cannot be sure that they're not dependent, and so we remove all but one of each one of those sets. And then we can trust that the reads are independent, and all our mathematical formulas in haplotype caller and all these programs works correctly. Does that make sense? The main problem that you need to worry about is when you have something like this, where you have an artifact that is duplicate, or you have maybe an error, maybe a true variant, we don't know, but it's been duplicated on many, many reads using from the PCR. And if we do not mark these as duplicates, this will look like, well, there's pretty good evidence for this. Right? But if we do mark this as duplicate, there's only one read that's supporting this variant, and so then we might not make the call. There used to be this thing called indel realignment. I'm not going to talk about that. It cleaned up indels significantly, but we don't need it with haplotype caller. So you may know that this thing exists, but we don't use it anymore. Finally, BQSR corrects the machine error. So it makes sure that we can take reads and we can say what is their actual quality versus what was the reported quality, right? And we can look at the differences. And so here's an example where we have we bin all the bases by their context, by their dinucleotide context, and we see the difference. This is plotted here. The difference between the reported quality and the empirical quality. You can see that the difference ranges about 5 Q scores. Right? 5 is quite a lot. It's half a decade. After recalibration, we get a relatively flat line that shows that the reported quality is very similar to the actual quality. There's various reasons why this could happen, but maybe we'll talk about that in the BQSR talk. Finally, for RNA-seq, we need a special aligner and a special step that will prepare the reads for GATK, because star emits these strange, for GATK strange, N bases in the cigar string that GATK doesn't know what to do with. These N bases basically say, even though the read continued from here to here, there's a junction, something was spliced out, and so it puts in back a N operator that says, just so that you know, these two bases were actually separated by, say, 1,000 bases of splice in an intron, and we splice that out. GATK doesn't know what to deal with that, and so to prepare the data, we pass it through this program called split and trim, which basically takes reads that look like this and splits them into two reads. One read is going to be over here, the other read is going to be over here. We're going to call variants over here, we're going to call variants over here. We don't need those two reads to be in the same place. that make sense? That's for RNA. We don't need that for DNA. All right. So I think with that, we go to the next step, which is variant discovery.